Okay, guys, so we talked about the basic cell structure, the function of each individual organelle. Now what I'm going to do is I'm going to get a little more specific about the structure of DNA. So let's take a little look at this video here, and I'll do a little explaining for you. All right, so look, any cell that is capable of dividing will have a nucleus, and it will have DNA. And DNA in every cell of your body contains all the necessary genetic instructions to make every protein in your body. And remember that you have structural and functional proteins, so they can make hormones which carry on all the chemical reactions inside your cells. This is the important piece. Regardless of the cell that it is, skin cell, liver cell, pancreatic cell, if that has a nucleus and it contains DNA, it's going to contain all the genetic information to make all the proteins in the body. Now this is the important thing. A liver cell only needs to make liver proteins. It doesn't need to make pancreatic proteins. So there has to be a way for chemicals from the blood to stimulate, let's say, a liver cell or a pancreatic cell to make their particular proteins. And how a cell gets signaled from chemicals from the blood are based on the type of receptors that are placed on the cell membrane's surface. So these cell receptors for different cells are going to be different. This is what allows certain chemicals from the blood to affect some cells and not affect others. And what are those cell receptors mostly made out of? Protein. So let's talk a little bit about DNA and how it works. First of all, DNA is the biggest molecule you have in your body. It's huge. And it is made up of these organic compounds called nucleic acids. Not amino acids, nucleic acids. And there are basically 4 kinds of nucleic acids. 5, but 4 right now. One is adenine, guanine, thymine, and cytosine. And when you chemically bond those nucleic acids together, you form a strand of DNA. Now, the DNA, you don't want it to get damaged. You get a genetic mutation. That's bad for you, right? You'll end up maybe with an eye in the back of your head, or worse yet, a genetic mutation that forces you to read the textbook. And that's not even close to being good, right? So here's another little important thing, and this has clinical implications in nursing. Adenine and guanine are nucleic acids that are referred to as purines. Thymine and cytosine are pyrimidines. Now, this is important when you talk about gout. Explain that later. So, if you chemically bond these individual nucleic acids together, and I'm making up this order, you got me? I'm making it up, right? Watch, making this up, right? You're going to make this strand of nucleic acids. This single strand of DNA is very unstable. It can be damaged easily. So the body always does stuff that makes sense. So what happens in a big, long strand of DNA, if you take, see, I'm making this up, A, T, G, C, T, A, right, C, C, C. This single strand of DNA is very unstable. But the body does stuff that makes sense. So to make sure that the DNA is much more stable, less likely to get damaged, there is a second chain of amino acids. That's what makes up that ladder-looking effect of DNA. And there is obligatory base pairing that goes with this chain and then the obligatory base pairing. This is very important. A always matches with T. So adenine always matches with thymine, and cytosine always matches with guanine. So here's one side rail of the ladder, and then when you chemically bond those base pairs together in that obligatory fashion, this little hydrogen bond forms the rung of the ladder, and A always matches with T, T always matches with A, G always matches with C, C always matches with G. So you match them up. And when you do that, what you will do is you will form this two-chained complex, this thing that looks like a ladder. And when the charges on the two rungs of the ladder interact, it will actually twist, and that's what allows DNA to get into that double helix shape. So again, what do you need to know? The 4 nucleic acids that make up DNA. The obligatory base pairing that goes into making DNA a very stable molecule, less likely to get damaged, that's good for you. And the obligatory base pairing, always A with T, C with G. Now, what's really important to know is this. Only one rail of the DNA ladder, or DNA staircase, whatever, codes for proteins, all the proteins in the body. Codes for other stuff, but in this class, it codes for proteins. The other rung is simply garbage. Its singular purpose is to make sure that DNA is stable. So let's look at this little video, and as we go through the video, I'll explain a few more things. Now, this sounds like a good question too. All right. DNA comes in basically two styles. It comes in packaged DNA, or in the loose, stringy form. The packaged DNA are called chromosomes, and we as human beings have 23 pairs of chromosomes. You get 23 chromosomes from your ma, 23 from your dad, and when they come together, you have 23 pairs of chromosomes. 22 of them are referred to as somatic. What does that mean? They code, they hold the genetic code to form all your physical characteristics. How tall you is, how smart you is, color your hair, color your eyes, stuff like that there. The 23rd chromosome is the sex chromosome. So if you remember from chromosome, yeah, I got it. It's right there too. Anyways, if you look at that little Punnett square, remember, men are XY, women are XX. So if you do the little square, right, XX woman, XX woman, XY male, XY male. So you have about a 50-50 chance of having a boy or a girl. Now look, because women both have the X chromosome and men have the XY chromosome, because they have different chromosomes that make up their sex chromosomes, the man determines the sex of the child. The woman determines when the man will have sex. You should write that down. Now, loose stringy forms of DNA, before I end that, let me say this. The only time that you see chromosomes inside a cell is when the cell is actively dividing. So only in dividing cells will you see chromosomes. Now look, I'm going to try and simplify this. Watch. If you got junk laying around your house, if you're living there, that's cool, right? But if you have to move, what do you have to do? You got to pack your stuff up, you got to label the boxes, and then you got to kind of organize it so you know what's going on. So when the cell is on the move, when it is dividing, it's got to take that loose junk that's all over your living room and kitchen, and it's got to package it up. So packaged DNA are in the forms of chromosomes. And the only time you see chromosomes inside a cell is when a cell is actively dividing. So if a cell is not actively dividing and you look in the nucleus, you're not going to see any chromosomes. What you will see is loose unraveled DNA ready to make some proteins. And loose unraveled DNA ready to make some proteins is called chromatin. And I'm simplifying this a little bit, but that's okay. So that's the difference between chromosomes and chromatin. Now, let's get into this a little bit. Remember, DNA, big molecule, made up of those 4 bases, adenine, guanine, thymine, and cytosine. So you look here at, huge, long DNA molecule. We are looking now at a section of DNA. This is important. A section of DNA that codes for a particular protein, that tells the cell how to put together the amino acids to make the protein, not only the type of amino acid, but the sequence of amino acids. That section of DNA that codes for a protein. Is called a gene. And in the human DNA, we have over 25,000 different genes that tell the cell how to put together proteins. It does other stuff, but for this class, it tells you how to put together specific proteins. Now, let's look at this a minute. As you can see here, you have this strand of DNA, and these little building blocks, these guys right here, these are the nucleic acids. So I'm making this up. This is A, T, C, and G, right? It's making up this part of the ladder. What's the corresponding base pairs for the other side of the ladder? A always matches with T, T always matches with A, C always matches with G, and G always matches with C. And so it's going to form this rung of the ladder. And remember, only one side of the ladder tells the cell how to put together the amino acids to make the protein. The other side is simply garbage. It's there to keep it stable. So in order for the cell to get the recipe to make a protein, you have to break the bonds between those 2 chains. And when a cell is going to make a protein, it cleaves those bonds between the A's, the T's, the C's, and the G's, and it reveals the 2 rails of DNA. So watch. And a big thing that looks like a turd, that's an enzyme, will come in and it will break the connections between the A's, the T's, the C's, and the G's. So in this video, it is showing that this rail rung of the ladder, right, this portion of the ladder, this tells you how to put together the proteins, the code for it. And then this side is simply garbage to keep it stable. Now look, DNA never leaves the nucleus, ever. So when a cell needs to make a protein, it is going to activate the gene, that section of DNA that codes for that particular protein. Now remember, you have all the genetic information to make all the proteins inside every cell of the body. But let's say you want to make insulin. And this section of DNA shown in this little video codes for the protein insulin. Would it make sense to unzip the entire strand of DNA, exposing all these little bases where they can get damaged? Or would it make more sense just to unzip the section of DNA, the gene that codes for the protein insulin? Well, the second one makes more sense. So that's exactly what happens. So when a cell, like a pancreatic cell, needs to make insulin, you got to get the recipe. Where do you get the recipe from? You get the recipe from DNA. So DNA is in a double helix to keep it stable. So you have to break the bonds between the A's, the T's, the C's, and the G's and reveal one of these chains of nucleic acids. And this bare naked chain codes for all for the protein insulin. Again, the other side is just for garbage. So I'm going to leave that alone right there. We're going to end this little short video, and then I'm going to talk to you about the process. I'm going to write this down, of transcription. Oh, that's too big. The process of transcription. Now look, if you copy somebody's test, what are you doing? You are transcribing it. You're copying it, right? So what you have to do in order to make that protein is you have to copy that section of DNA that codes for the protein insulin in this case. So DNA never leaves the nucleus. So there is a different type of nucleic acids chemically bonded together that will copy that section of DNA that codes for the protein insulin. So transcription is carried out by this stuff called mRNA. The M stands for messenger. So it copies the section of DNA, and now it carries the message to the rough endoplasmic reticulum, and specifically the ribosome, that will tell the cell, okay, buddy, this is the code that is needed to string together the amino acids to make the protein insulin. So next we'll talk about transcription, and that involves copying the section of DNA that codes for the specific protein. All right? Okay. Hope that went okay. Right?